Hello guys, welcome to the incredible world of insects. These tiny powerhouses are essential to our ecosystems, economies, and health. From pollinators boosting our food supply to disease vectors shaping public health, understanding these creatures is crucial. Join us as we share the activities we did in our entomology class, starting with insects-based morphology to trying to identify and classify them.
Let's begin by familiarizing ourselves with the basic external morphology of insects. Like all arthropods, insects have bodies divided into three main sections: the head, thorax, and the abdomen. The head contains the antenna, mouthparts, and eyes. The antenna vary greatly among insects but follow a basic structure. Segments 1 and 2 are termed the scape and pedicel. The remaining segments, called flagellomeres, collectively form the flagellum. These structures function in sensory perception, aiding insects in navigating their environment. Insects also possess various types of mouthparts, which can be grouped into two main categories: chewing and sucking. While all insects follow a basic mouthpart structure that includes the mandible, maxilla, labrum, and labium, these parts are often modified or reduced based on the insect's feeding behavior. Finally, the eyes. Most insects have compound eyes composed of numerous tiny lenses called ommatidia. These provide a wide field of view and are highly sensitive to movement, enabling insects to detect predators and prey efficiently.
And the next one are the legs. The legs of insects contain segments such as the coxa, trochanter, femur, tibia, and tarsus. Some insect species do not take the legs into account for their identification, but some, like the suborder of Rhopalocera or butterflies, can aid in the determination of their respective families. Moving over is the wings. This is one of the evolutionary advantages of subphylum Hexapoda and is primarily used for flight. In terms of identification, different genera can be classified by observing wing venation as well as the patterns such as the identification of Lepidopterans, as seen here with Melanitis vida. The last one is the ovipositors, usually a knob at the end of the last segment of the abdomen, purpose for laying eggs. Some insects, such as katydids, possess a prominent ovipositor. Meanwhile, other insect groups have modified ovipositors, such as order Hymenoptera, which includes ants and bees. Non-matriarchal individuals utilize them as stingers.
After collecting insect specimens, we will be needing the following tools. First one is the net, which is used for specific capturing these insects, and they come in different sizes. Second one are baits, which baits are used in order to attract insects, and these baits can either be starchy or sugary foods, which insects are attracted to. Next is the closure, which is used to kill the specimen with damaged bodies that we can observe the insect. Next are the forceps, which are used for grasping the specimen, but actually be careful not to grasp the specimen too hard to avoid puncturing or damaging the body of the insect. Are containers, which containers can either be small boxes, small envelopes, or even a ziplock to store the specimens after killing them. There is a paintbrush, and the paintbrush can be used in order for the small insects to adhere to its surface so that we can collect them easier. Next is the alcohol, which is used to preserve the specimen but can also be used to stun certain insects such as ants and mosquitoes that can easily be stunned once sprayed with alcohol, which makes them easier to collect. Next is the use of a GPS, which is for annotating the location where the insect was collected. Last but not least is the notebook, where we jot down the information that we need after observing the insect. We look for the materials that we need. Next is the collection process.
For collecting arthropods, we often use baits to attract them. For this demonstration, we have the mixture of peanut butter, tuna, mango beans, and flies, and place them into different locations in NRH building. The first one is the smiley face and the SP2 NRH bridge, which we use the sweepnet method, and the trees near at the court, and NRH cafe, which we use a handling collection method. 30 minutes have passed we collected more arthropods outside since the diverse ecosystem support their survival compared to indoor location. According to Worldwide Fire 2023, they maintain the healthy soil, nutrient, recycle, and pollinate flowers and crops. We also record the coordinates of our collected species, as you can see here in the figure. And now we will now proceed to our identifying our collected species based on their external morphologies.
In classifying and identifying insects, understanding their morphological characteristics is important as it provides insights for biodiversity, ecological, agricultural, and many more scientific studies. In our activity, we were able to identify mosquitoes and ants up to their subfamily and genus levels respectively. Identifying them up to the species level would require more than just looking at their morphologies, and further examination is required. By looking at the forward-projecting proboscis of this mosquito, we can tell that it can either be a Colicinae or Anopheline mosquito. Next, we look for its antennae. Since it is plumose or feathery, we can tell that it is a male mosquito. Then, we observe the length of its palps. For this mosquito, its palps is as long as its proboscis and it is not swollen at the ends. Combining this information, we were able to identify this mosquito as a male Colicinae.
Based on our last experiment, we discovered that our genus is Paracticina. To observe the morphologies of the ant more closely, we need to use tools such as insect pins, pinning block, and forceps. While pinning the ant, we need to use insect pins and a pinning block to attach the ant to the pinning block. The ant should be gently picked by its middle section positioning in the block. And while pinning through the middle section, we need to slightly arrange it to the right. The ant should be positioned correctly, labeled correctly, and most especially, it must be left to dry.
Thank you guys for joining me to explore insect morphology and collection techniques. If you found it helpful, don't forget to give this video a thumbs up. Want to dive deeper? Check out the cool links below for more amazing and mosquito mind-blowing video. Thank you.